Welcome to the start of a new system and what will probably be the most technically in-depth system that we'll wind up covering in AMP1. We're going to be focusing a lot on the physiology of the nervous system in the lecture. You'll handle the anatomy of the nervous system in lab, so we're going to focus quite a bit heavily on the functions of the neuron and getting much deeper into the topics of membrane potential. It's going to be important for us to understand the structure of a neuron, and I'll probably make this point again once we get some diagrams up in front of us. I'm not as interested that you know the structure of a neuron. However, the neuron is broken up regionally, and those regions are critical for understanding the function. So really, when it comes down to it, we'll need to know the structure of a neuron in order to get to understanding its function. We introduced the concept of membrane potential when we were in our membrane transport section. We are going to be taking that to the next level for neuro. Really, the entire function of a neuron is based on various ions fluxing back and forth that membrane, therefore disrupting that membrane's measured voltage. We're going to be taking various changes in that voltage and reclassifying, giving it brand new terminology and giving it a true in-depth part of the function of a neuron. We'll understand how one neuron creates connections with another, called a synapse. And while discussing that, we'll also talk about the actions at a neuron that bring about changes in an end organ. So first off, let's talk about the nervous system as a whole. It's an excitable system that is used primarily for controlling other systems. We had the overall theme of our AMP 1 and 2 classes as being homeostasis. And if you remember, we talked about how we maintain homeostasis. We looked at it from a feedback perspective. Receptor, control center, effector. Those were the three generic components of any feedback loop. Well, when we look at the different components, we can see the nervous system's fingerprints all over that particular system. Receptor, control center, effector. And of course, there has to be communication in between each of those three items. The receptor component will essentially always be nervous tissue. They're nerve endings that are specifically designed to detect various changes. Chemical changes, they're going to be chemoreceptors. Mechanical changes are going to be mechanoreceptors. Temperature changes, those are thermoreceptors. Light changes, photoreceptors. Those are always going to be neural tissue. The information from those receptors is transported into our control center. That communication is always going to be neural tissue. The control center itself is almost always neural tissue. Then lastly, we have the communication between the control center and the effector organ. That communication can either be nervous or endocrine. And then, of course, the effector organ itself can be any number of end organs falling in any number of systems. But you can see the nervous system is a huge part of those feedback loops. So we're going to be relying on the nervous system to detect changes in both our internal and external environment and be the communication network by which we can respond to those changes. When we talked about nervous tissue in our tissue section, we kind of strayed from our hard definition of what a tissue was. We said a tissue was a group of similar cells adhered together, working together, carrying out a function. Great example of that was when we did connective tissue and epithelial tissue. When we looked at nervous tissue, however, we said we had one main cell that And that was the cell that handled the function of the nervous system. When we describe the nervous system as an excitable system, what we're describing is the neuron. That's the excitable cell. If you want to picture the nervous system as the wires in the walls, essentially, that goes through the body, sending these impulses and communicating and controlling, what you're describing is the neuron itself. So we have the neurons carrying out the function, and then we have a collection of what we'll call glial cells. Glial cells are supportive. They are there to maintain and support the function of the neurons. First up, we have our astrocytes. Astrocytes are going to be housekeepers. They are going to maintain the chemical environment around a neuron. Once we look at the neuron a little closer, we look at the ions that are fluxing in and out of that neuron, you should get a pretty quick appreciation that the neuron is extremely sensitive to chemical changes outside its membrane. So we want to have control over that extracellular environment. We are going to rely on the astrocytes to help maintain that environment. If we have cells that die in the vicinity of a neuron, spilling its intracellular contents out into the tissue fluids, we're going to rely on astrocytes to come in and clean that up. We have our microglia, which are our immune system cells of the nervous system. They're able to migrate through nervous tissue and act as any white blood cell would in any other tissue. They're just specialized for the nervous system. Next up, I want to group these two together, because in lecture, again, we're going to be talking about these more so functionally. Schwann cells and oligodendrocytes. Both of these carry the same, if not very similar, functions. They help the neuron transmit their impulses more quickly. When present, and if you want to picture the neurons like the wires in the walls, when present, we will see the impulses travel through the neuron more quickly if Schwann cells or oligodendrocytes are associated. The difference between the two is geography. We will see Schwann cells in the nerves of the peripheral portions of the body. Oligodendrocytes will be associated with neurons in the central nervous system. Central nervous system, brain, spinal cord. That's where we'll see our oligodendrocytes. Schwann cells, everywhere else. Functionally, you and I are not going to distinguish between one or the other. So you'll hear me often maybe say Schwann cell in a lecture. For all intents and purposes, I could have used either name. Schwann cell is a little easier to say 50 times in a lecture than oligodendrocyte. I'm sure you can appreciate that. So again, in here, you and I won't be distinguishing functional differences between either of those. And last up on the list, we have our ependymal cells. You may have already heard of cerebrospinal fluid. That's produced in these chambers deep in the brain and ultimately winds up in a cavity surrounding the brain and spinal cord. Ependymal cells line those cavities, those chambers deep in the brain, and they're going to be the cells that produce the cerebrospinal fluid. So here is our neuron. So we'll highlight the main parts, but remember, we're going to be coming back to these anatomical portions of the neuron as we go based on its function, because we will see certain things happen in one part of the neuron that then triggers things to happen in a different part of the neuron. So first off, I want to draw your attention to the cell body. You'll also see that referred to as the soma in some textbooks, S-O-M-A. And as you can already see, extending from that cell body, we have any number of projections or appendages. The multiple short appendages are dendrites. Those are all dendrites. And our generic drawing of a neuron is going to include multiple dendrites. Typically, a generic neuron will have a singular, longer appendage. That's an axon. Cell body with multiple short dendrites, a singular, long axon. At the end of the axon, we have probably the one piece of anatomy that'll have more names than any other piece you'll study. But you can see those little bulges at the tips. You'll see those referred to as axon terminals. This diagram calls them synaptic knobs. You'll hear them called axon end bulbs. Regardless of what we call those, and I'll usually refer to them as axon terminals, those are areas of the neuron that contain neurotransmitters. A neurotransmitter is a chemical messenger, and we're going to rely on those neurotransmitters to communicate with other neurons or communicate with end organs like the heart, the pancreas, blood vessels, the diaphragm, whatever the case may be. Linking the axon to the cell body, we get this tapered portion. And on almost all the anatomical drawings, you'll see it referred to as the axon hillock. I actually prefer another term, the trigger zone. I call it the trigger zone, and you'll see it referred to in other resources as well. The trigger zone is the tapered portion where the axon meets the cell body. And from there, we can almost draw a line right across that trigger zone. And this line is really why we say that the neuron structure is related to its function, because we will be dividing the neuron up into these two halves very regularly. When we talk about impulses going down neurons, we're talking about them proceeding down the axon. Well, whether or not that occurs will all depend on what happens north of the line, so to speak. Whatever happens in the cell bodies and the dendrites will dictate if we get an impulse that goes down the axon. And the name trigger zone should make sense because that's the direct part that determines what happens down the axon. If we get enough of a change up here in the cell bodies and the dendrites to change the voltage here at this trigger zone just enough, then we will get our impulse. We need to change the trigger zone enough to send an impulse down the axon, just like you would have to change the force on a trigger just enough to engage the rest of the mechanism and have a bullet fly out the barrel. The only other pieces on this that we have to identify are the Schwann cells. And remember, Schwann cells or oligodendrocytes, we're not differentiating. We only see Schwann cells associated with the axon. And on this diagram, the Schwann cells are these light blue cells that wrap around the axon. We don't see Schwann cells associated with the dendrites, only the axons. The Schwann cell is going to wrap itself around the axon and continue wrapping and wrapping and wrapping. What's occurring here is we are getting multiple layers of Schwann cell membrane. That continues until what's left is very, very little intracellular fluid, but multiple layers of the membrane. And at that point, we call that myelin. And if we look at how the Schwann cells will form this sleeve that runs down the axon, we call this entire structure a myelin sheath. The last piece of anatomy on here is only relevant to the Schwann cells, and it's the spaces in between the Schwann cells, where we can look in between the Schwann cells and see axon membrane. We can actually look in and see bare axon. Remember, the axon is an extension of the cell body. It's got that phospholipid bilayer, just like we see in any other cell. And we have these bare spaces in between the Schwann cells. Now, I'll be referring to those as nodes of Ranvier, but you'll also see them called neurofibril nodes as well. So we have our cell body with our numerous projections. The short, multiple ones are the dendrites. The long, singular are the axons. At the tip of the axons, we have our axon terminals storing our neurotransmitters. And where the axon meets the cell body is our trigger zone or our axon hillock. And then finally, we see our Schwann cells forming this myelin sheath down the axon with the spaces in between where we can still see the axon being our nodes of Ranvier. To get back to our analogy earlier of these neurons acting like wires and transmitting impulses, we will generally see a one-directional flow. And it will roughly be in this direction. Basically, the dendrites and the cell body act as the input. The axon acts as the output. That's the general direction of electrical activity we see occur in the neuron. So now let's go back and talk about membrane potential. This was introduced back when we did membrane transport, and these are probably the same slides you would have seen in your notes at that time. We're going to revisit it and expand on it from there. Remember that a potential in physics is a separation of charges. As a kid, I don't know if you ever did this, but I sure did. You take an old battery, you wonder what's inside, you cut it in half. And you see that inside that battery was various chambers, different chemicals in each chamber. What that battery did was it separated charges inside its structure. We see a separation of charges across the membrane of a cell. When you would take your battery and connect the terminals with a wire, you can measure a current. If we take our cell and put electrodes, one on the outside of the cell, one on the inside of the cell, we can measure a current, just like that battery. So we need to kind of look at this neuron, or the membrane, at least at first anyway, and think about it in terms of that battery analogy. Across the membrane of a cell, we generally get minus 70 millivolts measured, and we'll come back to that number before we're done. So, separation of charges, just like a battery. The analogy that I like to use is salty banana. We have 5 different ions that we're going to be concerned with, all of which contribute to this separation of charges and the creation of the voltage. 5 different ions. This salty banana analogy at least lists 3 of them. We all know that bananas are high in potassium. So we see, with our analogy, high concentrations of potassium inside the cell. Most of us even know that salt, table salt, is sodium chloride. We see high concentrations of both sodium and chloride outside the cell. So far we get this sort of scenario. High concentrations of potassium inside, high concentrations of sodium and chloride outside the cell. Now, the sodium and the potassium in particular, those differences across that membrane, where they're accumulated, are going to be maintained by the actions of the sodium-potassium pump. Every time we break down ATP at our sodium-potassium pump, we will pump in 2 positively charged potassium ions, and we will pump out 3 positively charged sodium ions. So the actions of the sodium-potassium pump are what gives us the distinct difference in concentrations inside versus out of sodium and potassium. But we also have chloride out here so far as well, right? So that's 3 of the 5. The other 2, we have negatively charged proteins and or amino acids inside the cell. There's a high affinity for negatively charged proteins inside that cell. That's where they hang out. Outside the cell, we see high concentrations of calcium ions. So now we throw those in on our diagram, and here are all the players. We have our high calcium, our high sodium, and our high chloride in high concentrations outside that cell. Inside the cell, we have our high concentrations of potassium and our negatively charged amino acids. And then we have our sodium-potassium pump contributing to the differences of the potassium and the sodium across that membrane. There's our separation of charges. High concentrations of some ions inside and or outside the cell. Now, go back to our battery analogy. Even though you know that you can get a current out of a battery, you can picture that as the battery sits in its plastic container on the shelf of the store, you know that no voltage is being generated with that battery as it sits there. But you connect both terminals with a wire, and all of a sudden, like magic, you get your voltage. Now, without knowing anything about the properties of metals and how they conduct electricity, even from the most basic layman's perspectives, as a kid, you could picture something's flowing through that wire. The creation of voltage is somewhat a dynamic, fluxing state. That's what we have to apply to our cell model now. Because just like your battery, you don't actually get your current until you connect the terminals with a wire, and now something's flowing through that wire. We need to look at how these ions move back and forth this membrane, giving us the voltage that we're trying to measure. So we have to look at the movement of these ions and how they flux back and forth across the membrane in order to understand how it's going to look more like that battery with the electricity flowing through the wire. Now, to do this, we don't need to look at all 5 ions at once. As a matter of fact, that would just make it infinitely more complicated and abstract. So what we're going to do is we're going to simplify our scenario. We're going to look at one ion and watch how it moves. Then we're going to take that concept and envision that that happens with all 5 of the ions. So let's look at potassium. Remember potassium, salty banana, the banana part. High concentrations of potassium inside the cell compared to what we see outside the cell. Sodium-potassium pump keeps pumping in potassium. Now, remember when we did membrane transport? Even though we discussed all these different types of transport, we said that they don't occur in isolation. Active transport creates a concentration gradient, but a concentration gradient is the force that drives passive transport. So if we're actively transporting something across the membrane, we have to keep doing it, because as we keep pumping it in that one direction, it builds up a stronger concentration gradient, which only makes it leak back in the opposite direction more quickly by diffusion. Like having a leaky rowboat. You're in the middle of a lake, your rowboat is leaking, you've got a bucket in your hands. You're bailing that boat out. You're actively pumping water out of that boat, creating the concentration gradient, allowing water to leak back in through the cracks in the hull. So we may be pumping potassium in using our sodium potassium pump, but by creating this concentration gradient, that potassium is always going to be fighting diffusion in the opposite direction. It is going to diffuse back in the opposite direction through what they call potassium leak channels. Great name, because it tells you exactly what it does. You pump the potassium in, it leaks back out. Now, fortunately, you're pumping it in quicker than it's leaking out, so you can still accumulate a nice concentration gradient here. Now we have to start adding more things. What does positive and negative ions like to do with each other? Hopefully you said attract. Opposites attract. That's where that phrase comes from. Positive and negative ions like to attract together in ionic bonding. What else did we have inside that cell? We had negatively charged amino acids. So every time a positive potassium leaks out, it's going to leave behind an unpaired negatively charged amino acid. It leaks out, negative charge left behind. A second one leaks out, another negative charge is left behind. A third potassium leaks out, a third negative charge is left behind. What's occurring here is that we're getting potassiums accumulating outside the cell and unpaired negatively charged amino acids left inside the cell. There's where our attraction is going to come into play now. At some point, we'll lose enough potassiums and leave behind enough unpaired negative charged amino acids that the force of all of these negative charges are going to start pulling that potassium back across the membrane again. That force that pushes potassium out of the cell, down its concentration gradient, is the concentration gradient. The force that pulls it back in the opposite direction in this case is called an electrochemical gradient. Now we let that go into equilibrium. If this continues, at some point it's going to settle out. So now the rate that potassium leaves is the same as the rate that it enters back in. Now we've got, or what should look like, our current. There is our current, our revolving door of potassiums, leaving the cell, re-entering the cell, leaving the cell, re-entering the cell, leaving, re-entering, leaving, re-entering. By doing that, we essentially have created the positive and negative terminals of our battery. And if you've ever looked on the side of a battery or tried to put them back in the remote, one of the first things you always do is look for the positives and negatives and line those up. Otherwise, the battery won't work. This movement is what creates the voltage. We separated our charges earlier so that we have distinct concentration gradients created. This on the screen right now is essentially the battery in its plastic wrapper on the shelf of the store. we connect the wire to the terminals now, and we get our constant revolving door of potassiums and leaving and entering and leaving and entering. That is the dynamic state that we need to actually measure a voltage across that membrane. That was potassium. Remember, we've got 5 ions in here total. All 5 are going to undergo similar fluctuations. So if we just take potassium first, we can use a mathematical equation to calculate what we call its equilibrium potential. That's the voltage created only based on the movement of the potassium. That equation is called the Nernst equation. Concentration outside the cell divided by the concentration inside the cell. You take the log of that division, multiply it by 61.5, and we get, voila, minus 92 millivolts. That is potassium's contribution to the overall cell voltage. Now, earlier, I said that typically minus 70 is going to be considered the voltage of a cell, or at least any cell we'll talk about this semester. We've got 4 other ions, each of them undergoing their own flux back and forth the membrane, with different concentrations. We can do the Nernst equation on sodium, chloride, calcium, and the negatively charged amino acids. They're each going to have their own individual equilibrium potentials. And even though the math doesn't work this way, you know what I mean when I say take the average of something. Not all 5 of those ions contribute equally to the overall membrane potential. But if we take the average of all the 5, then we get our minus 70 millivolts for the cell. Because those 5 are by far the big players. Now, if you notice, the topic at the top of the screen changed. We're no longer going to be referring to membrane potential as anything meant to be specific. The neuron is what we've referred to as an excitable cell. And changes in its voltage is a critical part of its function. So if we were talking about a non-excitable cell, a chondroblast, an osteocyte, then its membrane potential is its membrane potential. It doesn't rely on its membrane potential as part of its function. So that's as far as that discussion was going to go. But just like every other aspect of anatomy and physiology, every single minute change has to be classified, has to be given a specific term. So therefore, if the potential of a neuron is meant to change as part of its function, we've got to give specific names to each of those changes. So now, we won't be referring to this as simply the membrane potential. We'll be referring to it as the resting membrane potential. That means that we have a neuron sitting there completely uninfluenced by any other neuron. It's at rest. The voltage in that neuron, in that state, is called its resting membrane potential. And that's what's going to be minus 70 millivolts. That is our more specific term now. We've kind of evolved the concept. So now we have to look at the different deviations, because we don't want a neuron that stays at rest all the time. We want it doing its thing. So we have to look at what changes the voltage. So in a cell at rest, we've got the sodium leaking out with its concentration gradient and being pulled back across to the outside by its electrochemical gradient. We've got potassium leaking out and getting dragged back in along its electrochemical gradient. We've got all of those movements back and forth relative to where they started and where they finish. That gives us our minus 70 millivolts. Well, what about if we opened up a channel? We put a protein channel here that allowed a bunch of sodium to enter the cell quickly. You should be able to perceive that that would throw off the equilibrium potential for sodium. Therefore, it would change sodium's contribution to the overall membrane potential. Therefore, it would change the amount of voltage we're actually measuring. And that would be 100% correct. Whenever you see the voltage of a cell changing, that is directly due to some degree of transport of an ion back and forth that membrane. So we've got three different channels that we're going to look at that are going to allow ions to enter and leave that cell, change the rate at which they flux back and forth naturally, therefore give us deviations from our resting membrane potential. The first type of channel is called a mechanically gated channel. Now, we won't really be discussing mechanically gated channels much, but how they work will allow us to better understand the other two. A mechanically gated channel sits in the cell membrane. And if that cell, if its membrane becomes distorted, if pressure is applied, if stretch is applied, if there's some sort of mechanical deformation of that membrane, mechanically gated channels open. Everybody listening to this lecture right now at some point has had experience with mechanically gated channels. You hit your ulnar nerve when you bump your funny bone. And you get a very distinct tingly sensation go down the medial aspect of the forearm and into the fourth and fifth fingers. By compressing that ulnar nerve up against the inside of the olecranon process, you mechanically have opened up the channels in that nerve. So mechanical deformation opens up mechanically gated channels. With me so far? What do you suppose will open up a chemically gated channel? If mechanical deformation will open up a chemically gated channel, hopefully somebody just said chemicals will open up a chemically gated channel, and you would not be wrong. It's like a lock and a key. You have a chemical present that acts as the key. You've got a channel in the membrane that will only open up if that particular chemical is present. Now, any number of chemicals can open up these chemically gated channels, but unless you're told otherwise, I want you to assume that the chemical we're talking about is a neurotransmitter. We mentioned that term earlier. Neurotransmitters are stored in those axon terminals of a neuron. These are the chemicals that open up these chemically gated channels. Now back to the regional parts of the neuron. Chemically gated channels are only present in the dendrites and the cell bodies of a neuron. Chemically gated channels are only present in the dendrites and the cell bodies of a neuron. The last type is the voltage gated channel. So mechanical deformation opened up mechanically gated channels. The presence of a chemical neurotransmitter opens up chemically gated channels. What would you suppose opens up a voltage gated channel? And with that, I can hear what you're thinking, and you're thinking voltage opens up a voltage gated channel. And that's not the whole story. We need a change in voltage. Not just any change, we need a big enough change. We need a change in voltage right around that channel that hits a certain amplitude. Then the voltage gated channel will open. These are only going to be located in the axons. Only located in the axons. Chemically gated channels were only found in the dendrites in the cell bodies. Voltage gated channels are only found in the axons. If a chemically gated channel opens, ions are going to move across that membrane, disrupt our equilibrium, and the voltage will leave the resting point. It'll change from -70. That change, for now at least anyway, we're going to call a graded potential. A graded potential. When chemically gated channels open, a graded potential is formed. And of course, this is only going to be in the cell bodies and the dendrites, right? Now, what is a graded potential? Your exams are graded. A measuring tape is graded. The speedometer on your car is graded. Broken up into small intervals that we can change at any given small interval along the way. So when your exam gets graded, you have the potential, the opportunity to get 0, 100, or any increment in between. They're variable. A graded potential is a small variable change, localized, right around where that chemically gated channel is. So you can have little tiny graded potentials, you can have slightly larger graded potentials, you can have slightly larger graded potentials again as you leave -70 millivolts, -69, -68, -67, -71, -72, -73, slight deviations away from our resting membrane potential as a result of the opening of a chemically gated channel is a graded potential. The other type of potential we have to concern ourselves with right now is the action potential. Graded potentials were small, localized, and created by the opening of a chemically gated channel. Action potentials are a very dramatic change in voltage with a more wide-reaching effect as the result of a voltage-gated channel opening. And you will see this drawing or something similar. If it's not 100 times the rest of this semester, I will be shocked. If we were sitting in a classroom with each other now, you would see me scribbling this on the whiteboard time after time. Action potentials are non-variable. A graded potential was variable. That's why it was called graded. An action potential is not variable. Every single action potential we will talk about will essentially be a rubber stamp. There is no such thing as a big one or a small one. It will go in a sharp depolarization pattern. Depolarization means we're going from resting towards 0. It has a sharp repolarization portion. Repolarization even means it's going from 0 down towards -70 again. It will have a hyperpolarization period, and then it will resettle back out. We will see that same pattern with the same degree of amplitude every single time we get one. Now you see here we have this dotted line, this threshold value. That's an important characteristic of our voltage gated channels. I said earlier they only open once we get a certain degree of voltage change around them. That is the threshold. Voltage gated channels operate as an all or none. We hit that threshold, they open up fully, or we don't hit that threshold and they stay shut. But as soon as we hit that threshold, we will get the rest of this pattern automatically. There's no stopping it once it starts. A toilet operates on an all or none. You jiggle the handle a little bit, nothing. You jiggle a little bit harder, still nothing. You have to hit a certain threshold of force on that toilet handle. Once you hit that threshold, as anybody knows who has flushed a stopped up toilet, and the panic that sets in afterwards, that once that flush begins, there is no stopping it. Just like once you hit the threshold here at minus 55 millivolts, the rest of this action potential takes place automatically and cannot be stopped anywhere along the way. So now, as we go, you're going to see we're refining our details, getting more and more and more complicated. So what I would suggest is after you watch this lecture the first time, you need to go back and watch it again. Because we're getting more specific as we go and leaving behind the simpler material as we go. So going through this one once simply won't cut it. Because all up to this point, we said that voltage gated channels create our action potential pattern. There's no way we could get that pattern with just one type of voltage gated channel. And in fact, we have two specific types. We have sodium voltage gated channels and we have potassium voltage gated channels. Working as a team in order to give us that repetitive action potential pattern. Each of those has its own threshold value. Each of them operate as an all or none. Those things aren't changing. And working together, we get our typical action potential pattern. So let's break down this action potential now and look at what happens with each of those types of voltage gated channels to give us what we see. The sodium voltage gated channels are the first ones to spring into action. And if you saw on the other diagrams, we said that the threshold to get an action potential was minus 55 millivolts. Well, secretly, that was for the sodium voltage gated channels. The threshold of a sodium voltage gated channel is minus 55 millivolts. Once we hit that threshold, sodium floods into the cell. And the voltage in the cell dramatically changes. And it will go from our minus 55 millivolts up to plus 30 as a result of that sodium flooding in. At plus 30, the sodium channels close again. And they don't just close. Boy, they slam shut. And they even have an additional protein called an inactivation gate. So not only does that channel shut, it locks the door as well. Once that inactivation gate is closed, there's a certain period of time when that sodium channel cannot be opened again. That channel now is completely inactive. So its job is to open at minus 55 millivolts, slam shut and become inactive at plus 30 millivolts. And it's the sodium that goes through the channel that gives us our depolarization phase, or our depolarization period of our action potential. So that's half done. The potassium voltage gated channels have a threshold of plus 30 millivolts. So it's the sodium that rushes through the sodium gated channels that hits the threshold for the potassium voltage gated channels. Potassium floods out of the cell quickly, dramatically changing the voltage of the cell, repolarizing it. But now, unlike the sodium ones, that as soon as they hit their endpoint, they slam shut quickly and lock the door, these potassium channels swing shut real slowly. So like one of those heavy doors on that pneumatic hinge. They start closing and they take their sweet time. Because of that, you get a few extra potassiums that sneak through while the door is closing, and we actually overshoot minus 70. We hit down here to maybe minus 80 or so. We call this dip our hyperpolarization period, until finally that potassium equilibrium is sorted back out and we wind up right back here at minus 70 again. So each of these changes in action potential is directly related to some action of those voltage gated channels. We go from minus 55 to plus 30 because that's when the sodium voltage gated channels open, and that's when they close, giving us our depolarization period. The potassium voltage gated channels open up here where the sodiums leave off. Potassium floods in, giving us our repolarization period, but they slam shut real slowly, giving us the hyperpolarization phase. And that's how both of those channels are going to work together to give us the full pattern of our action potential. So that's our graded potentials caused by the openings of chemically gated channels, and our action potentials created by the pairing of our sodium and potassium voltage gated channels. The graded potentials only take place in the dendrites and the cell bodies because that's the only place we get chemically gated channels. The action potentials only take place in the axon because that's the only place where we get our voltage gated channels. So now we kind of have to bring both of those together because we don't get graded potentials and action potentials occur in isolation. They work together. And in reality, it's the graded potentials that have to try and hit the threshold we need to create an action potential. This diagram here, remember I said we had our trigger zone. And I even drew a line across here and said what happens on this side of the line dictates if anything happens on this side of the line. And that's why we call that place the trigger zone. Well, we have a resting membrane potential in a neuron. If we provide a stimulus, we open up chemically gated channels, creating graded potentials in the dendrites and the cell bodies. If we can get that graded potential to change enough at the trigger zone to hit the threshold value for the voltage gated channels, then we will get an action potential formed in the axon that will then go down the axon and release the neurotransmitters from our synaptic end bulbs. We rely on the chemically gated channels in the cell body to bring about the change we need at the trigger zone in order to create our action potential. To look at that on our diagram, here's our resting membrane potential. We open up enough chemically gated channels to let enough ions cross that membrane to change the cell voltage. That's a graded potential. If it hits minus 55 millivolts, sodium channels open. We got the plus 30, potassium channels open. They start to close, they're closing more, now they're shut, and now we can reestablish our resting membrane potential at minus 70 again. So we've seen how we generate an action potential now, but now we have to look at how action potentials are regulated. Because we rely on these neurons and this nervous tissue as one of the most prominent communication methods throughout the body, we have to have a system in place to regulate the generation of these action potentials. Not only do we want to turn them off at times when we need them, but we also want to have a mechanism in place to make sure they're turned off forcefully when we don't. And that brings up the concept of inhibition. Remember, those action potentials are all or none. We hit threshold and we get one, or we don't hit threshold and we get none. When you are driving your car next, I want you to realize that you've got two pedals. One is excitatory, one is inhibitory. The gas versus the brake. Maybe you drive a stick and there's a third pedal. Forget that one for now. If you want to speed that car up, you can step on the gas and provide an excitatory stimulus, or you can lift your foot off the brake. In most cases, if you lift your foot off the brake, the car is probably going to start rolling a bit. So you can add an excitatory stimulus, or you can remove an inhibitory stimulus, both of which will get the car moving. Now that you're up to speed, if you want to slow the car down, you could apply the brake and actively apply an inhibitory stimulus. Or if you want to slow the car down, you could simply lift your foot off the gas and remove the excitatory stimulus. That's the way a neuron is going to function. Pushing on the brake is active inhibition. Most people are going to picture the brain almost like how you would picture a king or a queen on a throne, dictating organs to do what they want. Bicep, contract. Pancreas, secrete insulin. When the reality is, the brain as an organ is more like a single parent with 7 kids who are all hopped up on sugar, and you're walking through a store where everything is extremely fragile. And that parent is looking at the kids and they're saying, don't touch, don't touch, don't touch, don't touch. Actively inhibiting their actions. Well, what's going to happen the minute that one of them is out of sight and they're no longer being told what not to do? Well, that's when something gets knocked off the shelf and broken. You remove that inhibition. So if you want your bicep to contract, it's not so much that the brain tells it to contract, as much as it is the brain no longer telling it to relax. You remove the inhibition, contraction comes naturally. And that's why you see people with conditions like cerebral palsy, where certain areas of the brain have been damaged due to lack of oxygen. You lose that inhibition. Now they've got contracture of the upper extremities and unwanted muscle actions. So anyway, to get us back on track, look at how these graded potentials could change. And as long as we don't hit threshold, nothing's going to happen. Any graded potential that goes from resting and approaches threshold, we're going to consider excitatory. Any change in a graded potential that moves away from threshold, we're going to consider inhibitory. And we can go through those fluctuations, and as long as we stay under threshold, we do not get an action potential. So not only can we bring about a change in a neuron to get an action potential, but we can preemptively act on a neuron to make it more difficult to get an action potential by increasing the amount of inhibition. We regulate if we get an action potential or not based on what we do in the cell body and the dendrites to change its graded potential. Now, to take a step back, let's look at what we really mean by opening up a chemically gated channel. Because most of the time, this is a little bit of the chicken or the egg. Surely by now, someone has thought, where the heck do these chemicals come from to open up a chemically gated channel? And that's a good question. They also had to come from the axon terminals of another neuron. So somewhere along the way, an impulse comes down one neuron, releases neurotransmitters onto a second neuron, opens up a chemically gated channel, allows sodium to enter, and we get a graded potential. If we let in a little bit of sodium, it'll change and give us a small graded potential. We get more sodium coming in, we get a larger graded potential. We get even more sodium coming in, we get even a larger graded potential. If enough sodium comes in for us to hit threshold, then we will get an action potential. However, what about if another neuron is secreting a different neurotransmitter, opening up a potassium channel? And now potassium starts to leave. Remember, potassium is also a positively charged ion. Every potassium that leaves would offset the electrical effects of a sodium entering. So this could be an inhibitory action. This could be an excitatory action, giving us the opportunity to have these rises and falls underneath the threshold line. And from there, it's a tug of war. Is the excitatory stimulus going to be more powerful than the inhibitory stimulus, or vice versa, dictating if we get an action potential or not? So that opens up two brand new terms for us, which is exactly what you wanted to hear by now, right? But let's go back a little bit towards the beginning and see where we got here. It'll make these two terms fit in. We started with the generic term of membrane potential back when we did transport. Today, we got into the actions of a neuron. And all of a sudden, very quickly, membrane potential as a term became obsolete. And we introduced the term resting membrane potential. From there, we needed names to give to our changes in resting membrane potential. And we had graded potentials and voltage or action potentials. These are types of graded potentials. One is a graded potential that moves toward the threshold value. The other is a graded potential that moves away from the threshold value. We'll get into the terms of synaptic in a little bit. The synapse, to kind of give you a prelude, is the space between one neuron and another. So when you see the term synapse, that's what it's referring to. Presynaptic refers to neuron number 1. Postsynaptic refers to neuron number 2. Because remember, this is going to be a one-directional flow of electrical events. This is presynaptic, goes through the synapse. Now we're postsynaptic. So to go back and kind of rehash what we were talking about. These neurons are always being affected by other neurons. You got one innervating here and one innervating there and another there and another there. Some secreting neurotransmitters that are going to bring about an excitatory rate of potential. Others that are going to bring about an inhibitory graded potential. Whichever one of those wins will dictate what happens down the axon. If the inhibitory rate of potential is the stronger, then of course nothing will happen. We can't hit threshold with an inhibitory signal. And the remainder of this cell will stay just as it is, completely unaware of what's going on back here. However, if the excitatory stimulus gets strong enough and we get enough sodium entering that cell body, then we will change the voltage, hit the threshold, create an action potential. And once we create an action potential here, we will automatically get an action potential that spreads down the axon like dominoes falling. How it spreads is a topic for the next lecture. So for now, it's all about generation of an action potential, how we kick that process off. So it's important for you to understand how the ions move back and forth the membrane first. You have to understand regular membrane potential first. Then you have to understand how the movements of those ions contribute to the voltage itself. By doing that, you'll get the impression that if we change the voltage, if we change the flux of the ions even more, we'll have an impact on the voltage that's being measured. And that's how we can take voltage changes in the cell body, graded potentials, and translate them potentially into voltage changes in the axon, which would be our action potentials. And from there, we can control the impulses that go through the neurons and will give us a predictable communication device for all of those end organs that we need for our feedback control. So again, this is probably the most technical system we will do in A&P1. This is going to be the one system above all others that you're going to have to really work through and understand. Memorizing this physiology will not help you understand it. You have to put it into work. You have to discuss it. You have to understand how the system runs. So feel free to reach out. We'll discuss it as much as we need to, and I will answer whatever questions you have. And just keep working away on it, and it will start to make sense. Take care, and I will see you next time.